for joining us today, and a special thank you to our panelists for being here. Just a little bit of information before we get started. BioCanRx's Summit for Cancer Immunotherapy is coming up on November 21st to 22nd. It's a virtual event. You can go ahead and register for that for free this year. We'll be posting the information for that in the chat box for you to read and the links for you to click on to do that.
Great, thank you, Rita. And I'll just turn on my video here. I'd also just like to echo a welcome to our panelists. And before we begin, I'd really like to first make a land acknowledgement. So I'd like to start by acknowledging that BioCanRx is situated on the traditional unceded territory of the Algonquin and Anishinaabe people. And we acknowledge and respect our traditional hosts and thank them for allowing us to operate on this land. It's also important that you perhaps take a moment to acknowledge the land that you're on, and in the chat we'll put a link so that you can learn more about which land you're on, because of course we're gathering virtually, and of course it's different for all of you. And so with that being said, we are hosting this webinar on our first National Day of Truth and Reconciliation. And in order to engage in reconciliation, BioCanRx believes that we first must educate ourselves on the truths and roles that we have played in our colonial past and present. And so with that being said, I and we encourage you to spend some time to reflect on the atrocities of Indigenous peoples, and also to reflect on the incredible resilience of Indigenous peoples across Canada. So today you can engage yourselves in different ways. We recommend you reading the Truth and Reconciliation Report, educating yourselves on the land that you are on, listening to the voices of Indigenous peoples, and donating to Indigenous organizations. And so, like I said, I will post a link in the chat regarding whose land you're on. So with that being said, I would like to turn it over to Rita for the purpose of today's session. So Rita, take it away.
Thank you, Megan. So I'll start off by introducing very briefly our panelists today. So today we have with us Noelle Wilton, who is a research assistant at BioMarin Pharmaceuticals in the Manufacturing Science and Technology Cell Culture team. She has a Bachelor of Chemicals and Bioengineering from McMaster University. And during her degree, she actually completed 2 summer studentships with BioCanRx in the Guadalupe lab, where she focused on the development of novel purification methods for oncolytic viruses. She then transitioned out of the cancer immunotherapy space and began her work at BioMarin as an intern before joining the team as a full-time employee to focus on cell culture. Our next panelist, Sarwat, he completed his honors Bachelor's of Science in Biochemistry and his Master's in Microbiology and Immunology at the University of Ottawa. He then spent 5 years working at the Ottawa Hospital Research Institute, where he investigated T cell dysfunction and hepatitis C, and evaluated predictors of treatment efficacy of direct-acting antivirals against hepatitis C. He then went on to join Dr. Rebecca Auer's lab, where he worked on profiling the tumor immune microenvironment following treatment with oncolytic virus-based cell vaccines, and evaluated new treatment regimens to augment T cells and natural killer cell responses. He then went on to work with Dr. John Bell, where he developed immune assays to support different therapeutics being developed in his lab. He is now working at Nexelis in Laval as a senior scientist in the cell-mediated immunity department. And our final panelist, Mélissa Mathieu, started her undergraduate studies at Laval University before she obtained her PhD in immunology at the University of Montreal under the supervision of Dr. Nathalie Lebrec, where she studied the CD8 T cell immune response following vaccination. She then joined the lab of Dr. Simon Turcotte, where she studied the T cell immune response against neoantigens. She joined the development team at CellCarta as a senior scientist, where she now develops complex immunoassays for different sponsors on a variety of diseases and novel vaccines. So thank you and welcome to our panelists. Thank you so much for joining us today. We'll get started, and I'll just start off by asking the three of you to provide a little bit more background information and a little bit more information on yourself beyond what I've just given in your bios. And we'll start off from left to right, which would be Mélissa, Sarwat, and then Noelle.
Thank you for this introduction, Rita. So I did a really fundamental PhD where I studied the immune response, like and the antigen presenting cells. And afterwards, my postdoc, I wanted to have a more something that was more geared to clinic. So I went for translational. So it was kind of the best transition because I went from like working in a mouse model to working with human samples. So it was very great. I learned really a lot, but at some point I didn't feel fulfilled in that position. So I wanted to transition to the industry where I could work on bigger projects that were more achieved, let's say, that would be closer to the clinic. So that's kind of my scientific background. But throughout all of my studies, I always was implicated in organizing like scientific days, the student committees. This is something that is great and that it's all experience that I could bring with me now with in my official grown-up career. And I've learned a lot of skills in there. So what else to say? Throughout like my studies, I also had my kids, so I have all a family now. So I'm all in that like life balance that I can get maybe even more in the industry because we're part of a bigger team. So it's easier to help ourselves sometimes. So that would be it for my introduction.
Thank you, Mélissa. And we'll move on to Sarwat now.
Hi, I'm Sarwat. I think Rita mentioned briefly about what I've done. So I've spent a lot of my like training and career in immunology. So I started studying hepatitis C when I was at the Ottawa Hospital Research Institute with Dr. Angela Crawley. I was actually studying immune dysfunction caused by the virus's different proteins. And so that's how I got really interested in immunology. Initially, I was really interested in virology and HIV because that was very big back then, still is. So that's when I got introduced to immunology and I wanted to explore more about immunology. So I joined Dr. Auer's lab, where I got to profile all facets of the immune response to various therapeutic vaccines. A big thing at the Ottawa Hospital is the oncolytic viruses that Dr. John Bell had discovered with Dr. Soigel and Chio. And so we worked with a lot of those viruses to develop autologous cancer cell vaccines with Dr. Auer. So I evaluated those in preclinical models. I also was able to work on various systematic reviews of cell vaccines and other reviews on like NK cells and T cells. So it was a very exciting time for me to be at the Ottawa Hospital where I got to like teach myself a lot of immunology and kind of lead different types of projects. And so near the end, I did get an opportunity at Nexelis. So I moved here to kind of help establish the cellular immunology team. And now I'm leading one of the platforms here. So at Nexelis, we are a CRO, so we're a contract research organization that supports pharmaceutical companies in various phase 1 to 3 clinical trials. So in my career, I've kind of worked from like very basic discovery all the way through preclinical, translational, and finally now in clinical trials. So kind of like Mélissa, I've kind of gone through the whole process, and now I'm much closer to the end goal of therapeutics, where you're like now evaluating in real life populations and kind of figuring out how these vaccines will play out in the world. And very excitingly, with SARS-CoV-2, there's a lot of clinical trials going on, and we're constantly busy with like various SARS-CoV-2 trials, in addition to like Pfizer and Moderna that were happening.
Thank you so much, Sarwat. And we'll go on to Noelle.
Yeah, thank you. Great to hear a little bit more from both of you as well. I've never heard someone say excitingly thanks to COVID. So I guess that's just what you get when you sit in a room with a bunch of scientists. So my name is Noelle Wilton, and really Rita did a great job in summarizing kind of my experience with BioCan. I was really lucky to be a summer student in the Little Tulip Lab for 2 years, and that was kind of my academic stint. After that, I graduated with a degree in chemical engineering and biosciences, and I decided to not pursue further education. At the time, I just felt like I was ready to get out there in industry and see what else existed beyond academia for me at that stage. And so I made the move to San Francisco, California, where I work at BioMarin. And they are a biotechnology company, biopharmaceutical company that works in ultra rare genetic diseases. So a big pivot from the cancer space, but also extremely meaningful and exciting. And, you know, I feel like I've maintained that patient connection that BioCan and all of the initiatives that everybody here today is working on. And I work now as I recently transitioned into a process engineering role. So my background is in engineering, and I really wanted to move a little bit further outside of the lab still and look at kind of what we're doing in the manufacturing space and look at continuous process improvements. And fundamentally, you know, what I looked at even in my time at BioCanRx is very different than many of you with the immunology and science background. Engineers, our job is to take the brilliant science that you guys are doing in the lab and translate that to a scalable and economically feasible solution. And so that's really still what I'm trying to do in my current role. So thank you.
Perfect, thank you. And just a note for the attendees, if you'd like to ask a question to our panelists, you can use the Q&A function at the bottom. Alternatively, you can also use the raise hand function and we'll go ahead and unmute you and you can ask your questions. But for now, we'll move on to our moderated question portion. And so the first question I have for all of you, since you've all kind of transitioned from academia to industry, is what was your biggest challenge in making that transition? And I guess we'll continue with Mélissa, Sarwat, and Noelle for that.
I think one of the biggest challenges is because, you know, like at CellCarta, we're a CRO as well. So we work for many sponsors that will arrive with all kind of different projects. So in the academia, one part of it is a good side, but we're all on our own project. We know everything about it. We're the master of that project. But then when you go to academia, you're asked to be the specialist of a various kind of project that can touch different side of immunology. So to know what you need to do in all these little situations becomes very hard. Like there's a high diversity of what you're gonna face as scientific problem. And the company as a CRO, they often come to you when they're not able to perform an assay or to solve a problem. So you need to fulfill this need of them. But at the same time, it's very challenging and stimulating because when you succeed, like they're happy and you get to know different things and about a broader subject than like having like the only solution you have. So that was part of it, one part of it. And the other part I think is like understanding, because we're used to do everything because we're the master of the project, but then you do a part of the work. So it's a real teamwork, but sometimes it's harder to get the big picture. But I think like well, for me, I've been there for 3 years. So now I get more the sense of it, and I'm better at like getting the big picture and understanding what's happening. So that would be kind of the biggest challenge I face starting a new position in the industry.
Thank you. That's an interesting answer too. I never thought about it that way, that you kind of have to shift your way of thinking and perhaps even management. Sarwat, you wanted to say something?
Yeah, so for me, the transition from academia to industry, the thing that really popped out was regulations and the way we do GLP studies or GCLP studies and the level of GDP that you have to apply to all your studies. So in academia, you're often kind of like Mélissa said, doing your own project, you're writing things down in your lab book. But so when we're doing clinical trials, these are very regulated. The data that we generate are usually submitted to the FDA or Health Canada. So all of these, from the time the samples arrive at, say, Nexelis, to the time we release the data, everything is captured and regulated. Everything is written down. Everything is attributed. Every single reagent you use is cataloged and maintained. Every step you do is cataloged and maintained. So maintaining those processes, deviations and kappas and things that you get used to in the industry, those were very new to me when I first transitioned. And now like I navigate those all the time, every day for all projects. And like Mélissa said, different clients. So you have to kind of get used to that process, and the world of science kind of explodes in front of you when you join industry. And you kind of learn that it's not just about doing the experiment, it's about doing the experiment in the best way possible. And like Mélissa, like leading the team, it's important for me to ensure that the quality of the study is maintained throughout the process. So it's important for me to oversee all of these things and ensure that what we said we do it, and that the data we generate for our clients is of top quality. So I think that was one of the biggest, I guess, shock when you transition from academia to industry.
Interesting. So it's sort of like having to learn the business side of science, not just in the lab work. Oh, thank you. Noelle, would you like to add anything?
Yeah, going last, always so hard. But I think for me, I was very lucky. I did kind of my academic research, and then I did a year-long internship with BioMarin, the company I'm at now, and then went back to academia and then came back to industry. So I kind of was able to dip my toe in and transition as I, in a little bit less of a harsh way, I guess. No cold turkey for me. But I think really to echo what Sarwat was saying, everything is very highly regulated and very precise, and you have to make sure when you're making decisions that you're realizing the future implications. This isn't just on your publication or your study, but this has patient implications and implications on the whole company. And so it's really a lot of big decision making and really using all of those skills that you have and making sure you're utilizing everything that you have access to, not just your own brain or your immediate supervisor, but everything that the company has at their disposal. And I think that that's definitely a big transition, but I think all of the things that you learn in academia really set you up to make those decisions and to enter that space and have all of those skills.
Nice. So I think we'll flip the question now. What do you love most about your position? And you know what? We'll flip the order too. We'll go Noelle, Sarwat, then Mélissa, so that Noelle's not always last.
Yeah, we got to jumble it. Sarwat, you got the middle position. It's like the best. Yeah, what do I love most? I just really started in my new position as a process engineer, but I think in engineering, we're problem solvers and troubleshooters, and we always want to make things like the most economic and like the most efficient. And in my new role, I work on something called the process team. And so I support one specific manufacturing site, and I work on a team with like a manufacturing leader, a bioprocess specialist, so somebody who has expertise in the operations, a quality specialist, and a science expert. And then I am the engineering expert in that role. And together we really work to solve the major problems that are in our commercial operations and make sure that we're still meeting our target outputs and getting our product to our patients. And so that has like a lot of adrenaline. I feel like I'm such an adrenaline junkie and I'm always searching for that next big fix. And so this role really, really does give that.
For me, it's more being able to work with my team on a lot of different projects. As a CRO, we support a lot of different pharmaceuticals, a lot of different clinical trials, and we're able to touch a lot of different things at the same time. And as an immunology enthusiast who really, really enjoys immunology, T-cell immunology, NK cell immunology, I get to do that every day. And I get to do and apply it to clinical trials that are going to be used, like Noelle said, for like patients and directly applicable to others. So I really enjoy being able to do something I really like studying and like applying, and to do this with great people. So like working with wonderful people who make like everyday work like amazing, and we're able to do some great things. So that's kind of really what I like, to be able to kind of apply those things that I've learned in academia, and to do that with great people again is something that I really enjoy in my role.
So my turn. So at CellCarta, I'm in the development team. So I think the best thing we do is like problem solving, because we have to set up the method. So of course, like in academia, there's always problems. So we need to fix them. We need to make it happen. And it's always like, I would say also we're kind of adrenaline junkies, because there's always that first patient in that will push you to do it in a timely manner as fast as you can, because you need the assay ready for that first patient in. And as it's like biology, it gets always big experiment with often like, because we study like the immune response, and why do immune response like to be stimulated like 6 hours? It's always like, it makes your day terrible. But now since we're a team and a great team, like we all often work 2 or 3 on an experiment on a given day. So we make it happen. So it's kind of fun because we team up to make the project happen. And at the same time, we get all these scientific like question and problem and understanding what's happening, why it doesn't work. And when our assays are ready, it goes to the clinical department. But if they get problem for whatsoever reason, often it will come back to our table and then we need to again troubleshoot. So it's always kind of a back and forth and we ask questions and there's always that sense of urgency. And also it gives result kind of more quickly. It's not a long project over years that you will get an answer. Like rapidly you solve something. Like that week you solve the problem. Next week you've set up an assay. So I think I love that speed, the way things go.
Probably feels like you're actually getting stuff done instead of just
exactly.
sitting and waiting. Yeah. That's great. And going off of that, what does your day-to-day look like? And I think I'll let whoever would like to answer first answer, free for all.
Okay, I'll go. We're just gonna keep the order. For me, we start our day. I have a daily huddle that I do because I work on such a cross-functional team. We all get together and we look at the reports from BioMarin's manufacturing operation is 24 hours. So we look at the overnight reports, and then we look at what functional support they might need on the floor that day and kind of what our long-term trajectory is, and kind of base our decisions each day based on that. So my day can really vary. It can be these long-term kind of continuous improvement projects that we're working on, or it can be something came up overnight or the day before, and it's like, you need to work on this right now. So like what Sarwat said earlier, like a deviation or a corrective and preventative action, which is a CAPA, might come up, and I need to do that right away so that we can keep going with our process. So kind of like everybody said, it's a lot of that adrenaline of things need to happen now and feeling like you're getting things done because you have to get them done to keep going. So that's usually what my day looks like. Should I popcorn it? Melissa, you want to go in the middle?
I can go. So my days are kind of very different because as scientists, we do many tasks. So we plan the week ahead, 1 week at a time, and we have managers that will do this because they know what the priority are. So we each have given project, and in a given week, we'll be told to do this and that experiment because in function of what needs to be delivered, those first patient and the problems we're facing. And then who needs help? So we'll be kind of dispatched to help others. So I actually still go in the lab. So I physically bench experiment, but I also design them. And on other levels, I really kind of, now that I have more experience, I work, they call it the pre-contract discussion. So it's even before the contract are given. So some other days I will work on these, how can we make it happen, discuss with the client, what's available. So it's really challenging. And once the experiments are done, we also perform analysis. So I also do analysis to know, let's say if an assay is stable or whatever question we had. So I still do a lot of things. My position is as many tasks to be performed, while other positions sometimes will be more we have some analysts that perform regular analysis, people that are only in the lab. So it always depends. But often with masters and PhD, you tend to have those positions where you're asked to be more polyfunctional. So, but I like it. It's something there's a position for everyone, I guess, in the industry. People who like to have a routine and do the same thing every day, there's position for them. People who like to be more agile and have various schedule, there's other position for them. I have more of a funky, unstable position. So it's your turn.
Yeah. For me, it's a little different. I'm usually working on a lot of reports, either plans or reports for like clinical analysis plans or asset qualification plans and reports. I also like schedule my team. So like if there are assays that have to be done, so I will plan their schedule, I prioritize experiments. For client work, I'm also on client calls with my director to help them provide information on actual designs that we do and to support some of their projects. One of the things we do at Nexelis is we have the central lab for the Coalition of Epidemic Preparedness Innovations. So we actually train labs around the globe. So there are 7 labs around the globe that we're training for cellular assays. So we're involved in helping them develop their SOPs or reviewing their data and helping them set up different assays that they can support in different parts of the globe so that they don't always have to ship samples over to North America or to Europe to do their tests. So we support labs in Bangladesh, where I'm actually from. We support labs in India, Netherlands, Italy. So my day is very different. A lot of it is to do with like plans and report writing, reviewing data, writing reports, doing deviations and planning kappas. I don't often have to execute them myself, but like doing all of those reports. And I'm also in the lab to help the team like troubleshoot assays sometimes or to oversee analysis if they need some help with that. But most of my work is in front of a computer on a mouse.
So was that also pre-COVID? Were you also at home mostly doing work?
So, well, during COVID, fortunately or unfortunately, I've actually not had the fortune of working from home a lot. I do some, but I've mostly been actively engaged because even before at Ottawa Hospital, we were doing a lot of COVID research. So we're actively involved at Nexelis. We are very, like I live very close to work, so I go in and I oversee the work, and I was doing some lab work before as well. But I work from home from time to time, but I didn't get an extended break or anything of that sort.
That's nice. You get to sometimes go into work, sometimes not. Better than I think being stuck in one or the other.
Yeah, I think I didn't miss socializing as much because I got to go to work, and like Nexelis is fully staffed. So it's
nice. Got to see people. Yeah, that's a perk. So going off of what all of you have said, for someone looking to transition into industry, maybe from academia, what key skills would you recommend that they develop or really focus on?
Want me to go first? I think one thing I would tell someone who's starting out in science is to really get different experiences. One thing that I really enjoyed and really benefited me was doing my co-op terms during undergrad. I was able to work for the CFIA or even CFIA, Agriculture Canada. Then I moved to the Ottawa Hospital. So I would tell them to kind of explore different opportunities and not to stick to academic labs where you kind of work on a project and you're picking up skills, which is great, and you're doing exciting projects. But it's important to kind of try and see what other avenues there are in scientific careers, in terms of scientific careers. And it's important to be open-minded and to really kind of own your skills so you can like be really an expert at what you do so that you can apply that really well. For me, it was immunology and I really enjoyed it. So I really worked on it and I really focused on it. So I was able to progress that way. So I'd really recommend working on like finding something you really enjoy and work to get really good at it so you can be effective in what you do.
I think an important skill in the industry is your ability to communicate, to transmit your idea, to convince, because as always, if you have a project you want to push, you will need to convince like your coworkers it's a good idea, you want to change it, or your boss, and also communicate in like a gentle way so you don't frustrate anyone. The art of convincing people, the art of like asking people to do work for you, like in a nice way that people will want to work for you. Not necessarily as a management purpose, but just because you're gonna have co-workers, you won't be alone. Like there's few chance you will be working alone on a project. So it's gonna be in an interaction. So I think this is very important.
Yeah, if I may add, like, I think it's a great point for communication because as scientists, we are often used to talking to each other and using higher level terms, or so often when you're working in industry or any other field, really, you will not always be talking to someone who's studied the same kind of immune cells or same kind of like science as you. So it's very important to be able to articulate yourself, talk to different kinds of people, and obviously, like Mélissa said, you don't want to like antagonize anyone. So to be able to gauge others' responses to what you're saying is also very important. Sorry for butting in, but I thought that was very important.
No, that's good. You make me think like that. Even like vulgarization is important, because sometimes you'll be working with people from sample management or whatever. So when you'll have requests, like sometimes they will think it's odd or it's useless. So, but you have to like recognize this and be able to explain to them like, why is it that important for the assay? Because sometimes they don't have a scientific background. It's not that they want to do it wrong or whatever. So if you explain to them in a way they're going to understand, they're going to be more pleased to do it for you and do it well because they understand. So it's very important through all kind of the chain, the working chain, that people understand the importance of their task.
Yeah, of course. And, you know, so like, and actually transitioning from academia to industry, I think like what both of my panelists here have already said, I think it is really important to realize your audience and who you're communicating to, because often when we're in academia, you're working with primarily people who are focused on your very niche subject, especially people coming from like a master's or a PhD background. And so then when you're working with that broader audience, you definitely have to understand your audience before you start speaking about something, and make sure, you know, that can then achieve the goal, like Mélissa was referring to. And I definitely find that as somebody who works in the process side and the engineering and the development, maybe those, you know, niche scientific elements need to be translated differently to me so that I can reflect that in the process. And that's always something that I think is more amplified once you're in industry, because you are working with so many different people instead of just the people in your immediate circle and your immediate focus. So yeah, just emphasizing what they've said.
Oh, thank you. That's good. So what I've understood is communication to all different types of groups and people. No, thank you for that. And we actually have questions from the audience. So one person has asked, how did you make yourself stand out as an applicant when applying for the roles you are in?
I think it's hard to know what made us stand out. Like, in a sense, I was lucky because let's say I would have wanted to study for my position, I could have done the exact path I did. But at the same time, like, I applied for a position and they offered me another position. So, but like as a first position, when you're getting out of academia, it's sometimes very hard because you don't know what to expect. You're deemed as having no experience. Like, even if you have a PhD, a postdoc, you're just new to the industry. And in a sense, it's very true, because there's a steep step to understand. You talked about regulation before, all these things, these way of doing, the norms, the working in the GLP world. But I think it's where, when you have a lot of like side experience, like if you work in your community, past experience as a worker, not a student, you can use these experience to demonstrate that you can work in a team, that you'll be able to work in a timely manner. That's often things that matters, like, and demonstrating that you have a nice temper and that you can collaborate with others, because they always want people that will fit within the team. Like, if you have someone that has a bad mood or something that will like affect everyone, they will, even if you have all the technical skills, if your personality is not a fit. So it's hard, and at some point you need to be yourself. Because like me, they offered me a position that at the end probably fits me better than the one to which I applied. So be honest with who you are also, because you don't want a position you're not gonna like. for different reasons.
I also think, you know, I very recently went through the job hunt experience, and obviously I came directly out of an undergrad and not out of previous academic experience. But for me, it was really leveraging who you know. It's an incredibly competitive market now, and a lot of people have higher education and very impressive skills, and they're all applying for kind of the same jobs. So really leveraging your network, which, you know, if you're at this event and listening to this, you have such an incredible network, and use that to get out of what I call the pile. Like when you apply through a website or through the LinkedIn general portal, you are now in the pile, and your resume might be phenomenal, but there's no, like kind of Mélissa said, there's no support backing that. There's no who you are. There's no direct contact that makes that hiring manager say, I need this person. Because you all might have gone to the same incredible school and had years of academic experience and great internships, et cetera, and publications. But what makes you a person that they need to have on your team? And that's leveraging who you know. So me, myself, through BioCanRx, like I met vendors or different, you know, pharmaceutical and biopharmaceutical organizations who came to the summit. And also my PI knew people from when he did his PhD, who then ended up working in industry and were looking for people to hire. And so letting people in your network know, like, hey, I'm open to a position. These are my skills. I want to work at this company and this is why, can really help open a lot of doors. And then you get out of that pile and you become somebody that they need. They need you. They don't just need somebody with your background. They need you to be on their team.
Yeah, it's like
yeah, that is so, oh sorry, I went before you. That is so true. Like at CellCarta, they have a policy that they want the employees to send the resume of people they know. And I think like last year we hired 40% that were reference. And this is kind of, I think, the best foot in. Like if you know someone somewhere, like even if you know him or her just a little bit, like dare to ask if they would reference you.
Yeah, it's hard to tell what kind of sets you apart from all your other candidates that apply for a job. But like my colleagues just said, like it's important to highlight who you are and kind of leverage network. So for me, it's all about like tailoring your experience to the job that you're applying for. And often it's about taking the opportunity you get and kind of making more out of it. So not necessarily, it may not necessarily suit what you initially may have planned for, but then you can work on it to kind of achieve what you really want to get to at the end. You don't necessarily have to start at a certain level. You can start a little below that and kind of prove your way up the ladder. Because like Mélissa said, you could have had lots of years of experience in academia, but when you're applying to industry, all they see is that you just finished and you're just starting. So it's about leveraging that information, and it's really important to network, and it's great opportunities because people would like to hire people who they know are gonna work out and who they can trust that will work out. Like if someone I know is recommending someone, then I can be like, okay, so this person is more likely to work out than someone who I don't know being recommended. So I think it's important to network. And if anyone out there is an immunologist looking for a job, Nexelis is always hiring. So feel free to send your resumes.
We're gonna fight over with CellCarta for the candidates.
And those of you who don't know, Nexelis and CellCarta are both in Montreal area. We're not exactly in the same field. We're more in vaccines and more in immuno-oncology, but we are pretty much in the same.
Fighting for the same employees. And
yes,
we have a problem with the employees. Like there's a rarity of employees in every field. So in immuno-oncology and vaccine immunology too.
I think that's good news for anybody who's currently job searching. There you go, 2 options right there.
Yeah, I was gonna say my next piece of advice is to go to a job offer with a competing job offer. So here we go. Nothing makes you more desirable than having one of their competitors want you. So
No, these are all really, really great answers. Thank you. And we have another question kind of going off of mentorship and network. And so somebody has asked, you spoke about leveraging your network. What do you think are the best ways to actually grow that network?
I can go.
Yeah. Oh, you want to go ahead? Go ahead.
Sure. Well, BioCanRx is a great example. The conferences, the different seminars that they have, it's a great way to meet people across the country that are working on similar things. You may not necessarily, with the virtual networks, you may not necessarily see them, but you see what kind of things people are working on and what kind of things people are looking for. And that kind of helps you expand your network. It's important to stay in touch with people and make sure that you leave on good terms, no matter where you go or where you come from, so that you can always go back to them, or they may know someone that is in a great position in a company, maybe at BioMarin, that someone wants to join. And knowing Noelle through someone else might be the way to get into BioMarin or Nexelis or CellCarta for that matter. And I think through BioCanRx and through my work at the OHRI, interacting with a lot of immuno-oncologists around Canada, I've been able to kind of make those connections. It might not be solid, they might not be my friends, but I know them and they know of me. And that's the way to kind of leverage those networks. And don't be afraid to reach out. People are always willing to share advice and willing to help others. So reach out.
And probably like interest yourself to what your coworkers do, because we'll all be in the field later on. Like for the first position, that may not be like of great help, but you never know, like someone might be hired right before you, and otherwise, like all the symposia, as you just said, are great opportunity that must be seized. Like, of course, now it's more difficult, but I hope we're gonna get back to a real life soon, next year with BioCanRx.
Yeah, I think also like even though things are online now, like obviously not being in person is such a great opportunity to go up and like physically give your card or add somebody on LinkedIn right there. But maintaining the network, like Sarwat said, is so important. So you meet somebody and you follow up, you add them on LinkedIn or you send them an email and you maintain that network. You're like, you can't forget me, I'm right here. And you maintain that good positive feeling that they had when they met you. And then even when it's online on this webinar, right, everybody who's listening to this like should go and add all of the panelists and the other people that you saw in here on LinkedIn and send them a message and say, I really enjoyed you speaking, and this is what you said that spoke to me, and this is where I'm looking to go. And you make those personal connections, and then you might message me and I'm like, oh, I don't really have a lot of experience in that, but my friend that I graduated with is now working at this company and I can connect with her. And you just keep making those little personal connections with each person. And everybody in this space is so kind and has gone through what you're going through. And so I think has empathy and wants to help other people because they know how meaningful that was when they were in your position. So it's really just not being afraid and just taking those steps to do those things. And then a good other little cheat that I do sometimes is like, if you want to enter a space, you can search on LinkedIn, like a company or like an area of work. And then you can scroll down to the bottom and it's kind of like my network and who works here. And you can see like alumni from your university that now work at that company or in that industry. And then you could just message somebody like, I went to McMaster and I'm like, oh, somebody who went to McMaster now works at my dream company. Like I'm going to ping them because everybody has like that kind of like love for their alma mater and they want to help other people from that. So that's another good little cheat code that I use sometimes.
That's really good. Finding those little connections and using those to build on that. Thank you. Mélissa, you want to say something?
Yeah, I wanted to say there's some more general also like association, like Women in Biology, Biotech and Necto in Montreal. I don't know if it's like Canada-wide, that one, but they give like sometimes really great talk and you get to meet people from like many stages of their career. Like people really go to these like networking sessions for different reasons. And I've met like a good amount of very interesting person that never led to like necessarily employment, but it's contact that I have now for the future. And I know some friends who kind of by chance, they found their first position with these kinds of interaction, like someone leaving for maternity leave and saying like, it would be great if you want to try and have my position, you could submit your resume, like, which would have never happened without those like personal interaction. Even if luck was part of it a bit, there's always luck a bit, probably.
Oh, thank you. These are all really great responses. And so I have a question is, did any of you have any mentors? And if you did, you know, what was some of the really great advice that they gave you?
I can go first. Like, mentors are people that make you. Like, I've had great mentors throughout my career, starting with early on with Dr. Angela Crawley, or even before that when I was a second, third year student, kind of knocking on doors, be like, take me in your lab, give me a chance. Like, I first started working with Dr. Chris Body at the University of Ottawa, and I was a second year student with no experience. And I was doing organic chemistry a little bit there, a little bit of biochemistry, and that's where I started. And so all of these people kind of have a lot of influence on you. And then I went on to work with Dr. Crawley, was starting a new lab, and she really helped me kind of blossom as an immunologist, and like kind of giving my own project and trusting me to work on that. And then eventually I went to work for Dr. Rebecca Auer. Dr. Auer is a general surgeon, and she is a phenomenal leader, and she is always working for her patients, and kind of percolated throughout the entire team. So we're all very motivated by her passion. I've had great research associates who kind of mentored me during my time there. Dr. Michael Kennedy was one of them, and he kind of trained me to be a better scientist. And obviously I got to work with Dr. John Bell, who is really like a phenomenal character in biomanufacturing, biotherapeutics. So all of these people are kind of mentors for you, and they teach you something else and something new, and you always have to take the best away from them. And currently, even in my new role, my director has been very positive, and he's kind of encouraged me to take on more responsibilities and allowing me to progress in my career like I possibly couldn't do anywhere else maybe. So it's always about your mentors and about using their values, using their advice, and also doing for them. So making sure that you are someone they want to mentor. So I think that's very important. So I think for me, I've had a great opportunity to work with great people, and I really appreciate everything they've taught me. And I think it's important to acknowledge that no matter how far you go or where you go.
Melissa and I are like battling it out on who's going to unmute first. Game of chicken. Yeah, of course. There are, you know, I think I cheated and I looked at the Q&A and somebody was like, what did you gain from the BioCanRx studentship, Noelle? And I think some phenomenal mentors is one of the things that I definitely gained, you know. Without somebody who saw something in you, do you have the opportunity to then grow and foster abilities? And obviously there's a degree of putting yourself out there and searching for mentors. And I think that's something everybody should do is, you know, look for mentors and be like, hey, like, I think what you do is incredible and I want to learn from you. And that's kind of what Sarwat said, and making yourself open, making yourself mentorable. But for me, like Dr. Latulip, really, you know, it was like my second year of university, and he was like, hey, you know, I had a summer student last year with this BioCanRx thing, and I think that you would be a great fit. Do you want to read up about it? And I looked at it and I was like, I love this. This is in, you know, in a past life, I was like, I want to be an oncologist when I grow up. And then I grew up and I'm an engineer. It's kind of different, but I was like, this is so perfect. I'm going to get exposed to this whole network. And he really then pushed me to then question what I wanted to do and like the direction that I was going, and use my skills in ways that, you know, maybe somebody very green coming from high school, early university, they don't know the careers that are out there. And so that really helped me shape that. And, you know, having him as my mentor, getting this opportunity in the BioCanRx then allowed me to leverage all the skills I gained and get an internship at BioMarin, which then I got some other mentors who have really pushed me to look at my career differently and enabled me to then come back to BioCanRx and ask for something different in my next summer studentship, which gave me another pile of different mentors who had different skills, because I started doing more like cell culture things along with my purification background. And then I came back to BioMarin again with those new skills. So it's such like a ladder effect, and it's so like who you meet and kind of luck. Like Mélissa said, it's like, I'm so lucky I stumbled upon you, but then once you find those good people, like hold on to them and try and keep them close, because they're, you know, priceless for sure.
I have to tell like that during my studies, like my master and my PhDs, like I didn't have like mentorship. The kids are coming back from school. The dog is happy. I didn't have a mentorship like geared toward a transition to the industry. It was more kind of the opposite because I was in really fundamental immunology. And then at the postdoc, I had the chance to collaborate with the CellCarta and also with the Montreal Cancer Institute through a MyTac scholarship. So I don't know if you know of MyTac scholarship, but it's a scholarship that are meant to be a partnership between a student, an academic lab, and an industry lab. And there's a lot of like networking and soft skill training that goes along with those scholarships. So this is where I really kind of gained my mentorship for an industry transition by knowing people that had done it before. And because sometimes like the industry is seen as the devil, but it should not be. Like we're partners. We need academia to have like new discoveries for the industry. So it's not always easy to get that mentorship, but if you seek for it, you can find place like scholarship, networking that will give you that mentorship.
Oh, thank you. These are all really great answers. And we're already at 3:54. Like time just flew by. But Noelle, did you want to add anything to that last Q&A about what you gained from the summer studentship program?
Yeah, sure. I think, you know, it's such a great opportunity, obviously, to meet people. That's something I said, and gain incredible exposure. I can't, like the talks that have happened at the, like I went to the summit in Banff, so that was 2018, I think, and then in Victoria, is that 2020, not 2019, I don't know. All the years blend together now. I'm like, I've been sitting in this room for a decade, it feels like. So, and meeting people and listening to those talks were so inspirational at such a pivotal time in my career. So getting to go to that, getting to work, you know, I really felt in my lab that I was treated with like such respect, and so many opportunities were given to me to take a project in a different direction and try something out and publish, which maybe I'm grateful I don't have to do anymore now that I'm in industry, because the rounds of revisions ate away at my soul. But I think all of those things were so meaningful and really gave me, kind of going back to the resume thing, those tangible skills. So I got the network from this opportunity, which got me an internship, because otherwise I wouldn't have had any skills when I applied for that internship. And then I came back and gained more skills and things, and really was just given so many opportunities. So I can't speak highly enough to this summer studentship program. I think it's really important to give these kind of unique opportunities. And I think it's so impressive, the science that's happening in this space. And I think it's really impressive that so much of it is happening in Canada. I think for me, you know, I work in the US now, and the market here is very different, and the research opportunities and things like that. And I think BioCanRx is a standout opportunity in Canada to really develop a lot of skills and see that bridge from academic to industry in our home country. So I think it was a fantastic opportunity. I would go back and do it 2 more times if I could.
Well, that's great. Thank you so much, Noelle. And since we're almost out of time, maybe one last takeaway or piece of advice from Mélissa and Sarwat for all of our listeners.
Well, I think if you're thinking about leaving academia for the industry, like it's a must try. Like maybe it's going to be a good fit for you, maybe not. There's always like going back and great opportunities in different fields. So just try it.
For me, it's what I always say is like, find something you really love doing and work on excelling at it, and then keep trying. Don't keep pushing yourself, keep trying for something better, and the opportunities will come. So seize it when it does.
No, thank you so much, everyone. This was a great session. I appreciate all of your enthusiasm, and you all gave great answers. So on behalf of everybody here, thank you all so much for your participation today with us.
Thank you.
Yeah, thank you for having us.
Yeah, thank you so much.
And I would just like to echo that.
Some of you.
Yeah, and we will be posting this video to all of you just for your reference. Also, feel free to share it with your community. And just a final reminder that we are hosting a virtual summit for cancer immunotherapy coming up in November. We'd love to see you all there. It is free this year. So, so maybe you're in a different space now, but you're part of the communities and always will be. So hope to see you there. But again, I'd really like to echo what Rita said. Wonderful responses, both tangible, intangible advice. So thank you very much for participating.
Thank you. And see you at the summit, hopefully.
Oh, wonderful. Good. Excellent. All right. Thank you very much. Thank you all. Have a good day.
Bye, everyone.
Yes, thank you, everyone. Have a good day.
Bye now.